Welcome to this introduction to the regulatory world. My name is Harald Enzmann.
And my name is Sabine Mayhofer, and welcome to this introduction on the authorization of medicinal products in the European Union.
This is the very first part of our webinars on medicines regulation in the European Union. For starters, we will try to give you the big picture, the overview, to outline what drug approval is all about and how it works.
Slow down, Harald. We need to cover formalities first.
Thank you, Sabine. Indeed. We are both employees of the German Regulatory Authority, the Federal Institute for Drugs and Medical Devices. And we both work on centralized procedures with EMA. Therefore, we have to make this disclaimer. This presentation reflects our personal views. If it is inaccurate or you just don't like it, blame nobody but us.
In this presentation, we will outline what drug approval in Europe is all about, how it works, and how and by whom the scientific assessment is done.
We will start this webinar with the overarching goal, patients benefit, and then we will look how we do this in the European Union or the European Economic Area. We will try to introduce you to the admittedly complex European network with a particular focus on the CHMP, and we will explain the principles of our benefit risk assessment.
Let's start with the overarching goal of it all, with the ultimate purpose of drug approval. Originally conceived as a safety net, as a goalkeeper, it has now taken on an equally important secondary role of advancing the development of new medicines and making them available to patients. Originally, medicines regulation was meant as a safety net. Regulators were to be goalkeepers. A major milestone for this was the sulfanilamide catastrophe in 1937.
A sulfonamide syrup. In the pre-antibiotic area, these were the most effective antibacterial drugs. Such a syrup was spiked with diethylene glycol, causing the death of over 100 people, including many children.
It became obvious that pharmaceuticals do have an inherently higher hazard potential than almost any other goods or commodities.
US Congress responded by passing the Food, Drug and Cosmetic Act of 1938, which introduced for the first time the requirement of safety tests before a medicine would be allowed to be marketed. Other nations learned from that and developed their own national legal framework, some sooner, some later. It is concerning that even today, this first function of a medicines regulation, stopping bad medicines from damaging patients, the goalkeeper function, cannot be taken for granted.
It is inconceivable, but it seems nothing has changed since then. Even today, children die because diethylene glycol is in their medicine. But stopping bad medicines from damaging patients is only one side of a regulator's job. The other side of our job is to clear the way for beneficial medicines, facilitate the development from research to clinical routine.
Today, we are fully aware that the approval of a drug is not an end in itself. Allow me an analogy. The bridge from research to clinical routine. 5 pillars, 5 major decision makers, each with a different mandate, with different interests, with different decision criteria.
And the regulators in the middle, working with all of them. We will come back to this at the end of our presentation. But just for now, this message. Regulators, both as goalkeepers, stopping bad medicines, and as enablers, bridging from research to the patients, opening the path for better treatments. All regulators know that marketing authorization cannot be an end in itself. It is obviously bad for patients when a drug has been approved with a positive benefit-risk ratio, cannot be used in patients for cost reasons. And it is also frustrating for us as regulators when we have put a lot of work into this positive benefit-risk assessment, and in the end it remains useless. It was even futile work because the drug does not reach the patient. So in the end, we want to have an intact bridge before and after our own pillar. That's why we work together with other stakeholders and we try to support them.
We work with upstream decision makers, such as industry, giving them scientific advice for the mostly clinical development.
And we work with downstream decision makers, providing the first objective, impartial assessment of a new medicine, on which others can build their own assessment and decisions. HTA bodies and payers start with our assessment when they perform their own assessment of cost effectiveness and decide on reimbursement and pricing.
And with the public information we provide, in the SMPC and in the EPAW, we support patients and physicians when they make the final and most important decision, when they decide whether a particular medicine is the right choice for an individual patient. In the European Union or in the European Economic Area, what we call the regulators are organized in the European Medicines Regulatory Network. We will have a look at the two major components of this network. On the one side, you see the centralized European part with the European Medicines Agency and the European Commission. On the other side, you see the decentralized part of the network with member states and their national competent authorities. The EMA was never designed to become a monolithic block like the US FDA, but was always designed to lead a network of national agencies. As you see here, on the one side, we have these national authorities in the 27 member states of the European Union or in the 29 members of the European Economic Area. They have the responsibility for most of the generic marketing authorization, the authorization of clinical trials, and most of the medical devices. On the other side, we have the European Medicines Agency with their focus on the authorization of innovative medicines, pharmacovigilance, and referral procedures in case of conflict between the scientific assessments in different member states. The important difference between this European model and the US-American FDA is that EMA or EMA colleagues do not make the scientific assessment. An idea how this works in this network gives you the next slide. While it is the European Commission who in the end formally decides on a marketing authorization, the scientific committees here in the middle, in the center of it all, it is these committees where we develop our scientific opinions, where we define the criteria for the assessment, where we set the scientific frame. Doing this, the committees are supported by several working parties, by colleagues from the national agencies, in the European specialized expert communities, and by experts from hospitals and universities in the scientific advisory groups we have.
The important thing is that neither committee members nor working party members come from EMA. They come from the member states. Mostly they are members of the national competent authorities. EMA coordinates the decision-making bodies, coordinates the committees, and organizes the working parties. But the committee members who make the decisions in the end are not from EMA. Over the years, EMA has accumulated 7 scientific committees, although this may change again with the new pharmaceutical legislation. The centralized procedure for marketing authorization for medicines for humans is the responsibility of the Committee for Medicinal Products for Human Use, in short CHMP, in collaboration with the Committee for Advanced Therapies, in short CAT, and with the Pharmacovigilance Risk Assessment Committee, in short PRAC.
The composition of all of EMA's scientific committees is similar. One delegate from each member state, sometimes with, sometimes without an alternate member, and a variable number of co-opted members. We will focus now on the CHMP because this is a committee where applications for marketing authorization applications for new medicines will come in and will live or die there. We will first have a look at the composition of CHMP, how CHMP is organized, how the procedures are run at CHMP, and then we will see how CHMP members make up their mind on the benefit-risk balance of a new medicine. For CHMP, each of the 27 European Union member states nominates one CHMP member and one alternate member.
The 27 members elect up to 5 co-opted members, providing additional expertise. Members from the European Economic Area, from Iceland and Norway, fully participate in the work of the CHMP. The committee has regular meetings each month, where scientific opinions are discussed and finalized, in most cases by consensus. If no consensus can be reached, the scientific opinion that will be the basis for the decision by the European Commission follows the majority. A majority requires 17 out of 32, the 32 being the 27 EU member states' votes plus the 5 co-opted members. The committee members build their opinion on the assessment reports provided by the assessment teams of rapporteur and co-rapporteur, and on the support from experts of their national agencies from back home.
When a marketing authorization application is submitted in the centralized procedure, CHMP appoints a rapporteur and a co-rapporteur who will lead the assessment and, with their teams, draft the assessment report. Rapporteur and co-rapporteur are CHMP members who volunteer, and the selection of rapporteur and co-rapporteur is based on the expertise of their teams. In the beginning, each team, both rapporteur and co-rapporteur team, used to be composed of experts from only one national agency. More recently, though, there is a clear trend to multinational teams, with members from more than one member state, reflecting an increasingly close integration of the European regulatory network. We need to emphasize the importance of the rapporteur and the co-rapporteur team. These teams do most of the assessment work, and their conclusions clearly influence the final outcome.
On this slide, you see the overall timelines in the centralized procedure. We will not go into detail here. Procedural aspects such as the structure of the assessment report and timelines will be the topic of a separate presentation by Francesca Day and Martin Norta. For now, it will suffice to see what we have after the validation of the submission in blue on the left, a maximum of 210 days for the assessment, here symbolized in the red part. Usually with 2 or more rounds of assessment, that means questions to the applicant, applicant's response, and assessment of the responses. And then the European Commission receives the CHMP opinion for their final decision. Within these up to 210 days, the teams of rapporteur and co-rapporteur have a leading, sometimes dominating role. Most of the assessment time, here shown in the green, the light green, it is the rapporteurs and co-rapporteurs teams who work on the assessment, while other committee members and their teams, in their role of the so-called other member states, invest considerably less time, here in orange.
In every cycle, the rapporteur and co-rapporteur teams will prepare a draft assessment report. Other member states will comment on this. The rapporteur and co-rapporteur teams will include their comments or prepare a reasoning why they do not want to accept these comments. Therefore, unsurprisingly, it is rare that rapporteurs and co-rapporteurs' conclusions are overruled by a majority of the committee. After all, these teams know all the details of the submitted material. These teams spend the most time in assessing the material. And this is why it is important that these teams get it right in the first place. When we say that 210 days is the legally defined maximum time for the assessment, there is seemingly a contradiction to the much longer times actually passing from submission to marketing authorization. For example, 426 days according to complaints by the pharmaceutical industry. Now, Sabine, can we solve this apparent contradiction?
Yes, Harald, we can. And there's the explanation. On average, less than 200 days are used for the assessment by our teams, here in light blue. There is the need for a couple of days after CHMP opinion for administration, here in yellow. European Commission's time here is in green. It is remarkable that a big part of the overall time, about 160 days, is not at all used by the regulators for the assessment, but by the applicants during the so-called clock stops, when the companies work on responses to CHMP's questions, here in dark blue. Please note, in 2022, for the first time, the companies, the applicants, used more time for these clock stops for completing and amending their submissions after the assessments had started than we on the regulator side used for the assessment. Nevertheless, and no matter if we need too much time for the assessment or if the applicant needs too much time to amend and complete their submissions, overall, roughly 400 days from the start of the procedure to the decision by the European Commission is admittedly too long. As this has been stable at about 400 days for several years, many thought there could not possibly be an option for dramatically shorter procedures.
Well, the COVID pandemic winter 2020-2021 taught us differently. In a 78-day procedure, the first vaccine was approved. In a 53-day procedure, the second one. It has been quite amazing what was possible in this first crucial phase of the pandemic last winter, when we knew vaccines were our best chance to control the pandemic, and that indeed each day mattered, as every day a few thousand people were dying from COVID in Europe. It was amazing what the system's European regulatory network could do. There was in those times a willingness to drop some formalities and procedural niceties. For example, we encouraged the companies to submit data for assessment whenever they became available, complete or incomplete, and not as usual wait until a complete package was available. And there were assessment teams, literally, working 24/7, changing at 3 in the morning, with some going to sleep when others just came back online to continue where their colleagues left off after just a few hours of sleep.
But the success was worth the effort. Within 78 days, the first vaccine was approved, and within 53 days, the second. No emergency use deals with different national decisions all over Europe, and no constructs with marketing authorization holder and manufacturers dodging every responsibility. We achieved a regular European marketing authorization with complete assessment for benefit and risks, with benefit and risk assessed as in every other procedure, only faster. Bottom line, the European regulatory network worked exceedingly well in this unprecedented situation.
Well, yes, we did it, but at a price. Looking back today, I can say, no, these weeks were not fun. And partners and families suffered with the assessment teams and with the committee members. Nevertheless, if you'd ask any of them today, they will tell you they would do it again. The biggest crisis of our lifetime yet, and they were in the middle of it. And they did it just doing what previously was considered impossible.
Clearly, the experience of the pandemic has changed a lot, including our views of the limits of regulatory work. While the 50 or 70-day procedures for the first COVID vaccines obviously cannot become the new standard, it is equally clear that things can be done differently. The procedures can be streamlined and the assessments focused.
It can be done differently, and it will be done differently. What cannot change, however, is that the benefit-risk balance is and will remain the center of the assessment. Now, how do we assess? How do we evaluate the benefit-risk balance of a new medicine?
We may use this sophisticated instrument here, but usually we simply build the benefit-risk balance on the assessment of efficacy, safety, and pharmaceutical quality. This means regulators' conclusions or judgment on the benefit-risk balance of a medicine will be based on the medicinal benefits, and the medicinal risks of the patients or public health will be based on the assessment of efficacy, safety, and pharmaceutical quality.
Other important issues, such as the medicine's impact on the environment or on animal welfare, may be considered and discussed, but cannot change the benefit-risk balance, as the benefit-risk balance, CHMP's opinion, is based on Directive 2001/83, Article 1.28, first ident only. Well, at least in the current legislation, that might change, but that is certainly years away. For CHMP's opinion, for the recommendations for the European Commission to grant or not to grant a marketing authorization, the committee will balance the benefits for the patients, the favorable effects of the medicine, against the unfavorable effects of the medicine, the risks that the medicine will harm the patients. It should be evident that outstanding benefits, reliable effectiveness in a life-threatening medical condition or disease with no other treatment options may justify even significant risks, while the very same risks would be completely unacceptable for a medicine meant to treat a less severe condition, to treat an ailment for which patients have several other treatment options, say for a medicine for a mild headache. While safety may be relative, efficacy is not negotiable. If the medicine just does not work, then it will not help if the pharmaceutical quality is the best of the best. It will not help if the medicine has no adverse effects either. With other words, for most marketing authorization applications, efficacy is the most crucial criterion. Efficacy is a conditio sine qua non. If efficacy has not been demonstrated, this is the end of the story. This is a showstopper. And this is why we will now focus on efficacy. The efficacy of medicines is usually established by clinical trials. The gold standard, the ideal study, is the prospective randomized double-blind trial. The double-blind trial design remains the best protection against placebo effects. The best possible protection, albeit not a perfect one, as it may be unavoidable that the blinding becomes partially undone, for example, by characteristic revealing adverse effects of a medicine, which patients and their physicians may recognize. Prospective randomization prior to start of the treatment is the best possible protection against putative treatment effects that are nothing but selection bias. Randomization at the beginning of the trial is clearly the preferred way to do it. Later, even after the treatment, the pairing of seemingly comparable patients can help. But it can consider only criteria known as sources of imbalance, the ones we know, and criteria we think that they might possibly become a source of imbalance, the ones we know we don't know, the known unknowns. However, completely unexpected influences, the unknown unknowns that might cause severe bias, cannot be addressed. Of course, while the randomized double-blind design remains the gold standard, there are exceptions. Sometimes single-arm studies can provide the basis for marketing authorizations if they fulfill certain conditions. Here, I highly recommend a look at our latest reflection paper on efficacy in single-arm trials. If we want to base regulatory decisions on single-arm trials, the natural cause of the disease must be sufficiently well known, the treatment effect must be compelling, and the treatment effect must fulfill an unmet medical need in a severe medical condition, meaning the new medicine is either the first treatment that becomes available or it is obviously better than everything else available. Using a clinically relevant endpoint means the treatment effect is preferably measured by something directly relevant for the patient. For example, if the patient can climb stairs or is bedridden, if the patient lives or dies. Or it can mean something that may not be directly perceivable for the patient, but is generally accepted as a surrogate. Something that has been convincingly shown to be predictive for the course of the disease. For example, in diabetes, continuous HbA1c levels of 15% must be expected to have dire consequences, even if the patient is feeling well at the moment. HbA1c is a classical surrogate. On the other hand, we would not use Bence-Jones protein as a surrogate endpoint for the treatment of multiple myeloma. Not validated. Doesn't work. But speaking of proteins, there is of course a gray area. You may be aware that CHMP has been less convinced of the validity of amyloid as a surrogate for the efficacy of new medicines for the treatment of Alzheimer's disease. But CHMP has been hesitant to accept the quantification of the amyloid as basis for the marketing authorization decision, whereas FDA placed a high bet on the amyloid hypothesis. For the regulatory decisions on marketing authorizations, we will always come back to the one question: do we know with sufficient certainty that this medicine will make a perceivable difference for the patients? But even if we do not have a single-arm trial, if we have a control group, the choice of the appropriate comparator group can be crucial for the study design. For clinical conditions without established therapy, superiority to placebo should be shown to demonstrate efficacy, unless, of course, randomization to a placebo group would be unethical. It is more complicated for medical conditions with existing therapy options. When the new therapy is intended as an add-on to an existing therapy, it again is a comparison to placebo. Patients in both groups will receive the previously established standard therapy, on top of which one group will receive the new therapy and the other group will receive placebo. The study will then show whether the new treatment adds any benefits to the standard treatment. If the new treatment is intended as a potential alternative, not as an add-on, alternative to the established treatment, one group will receive the new product, the other group's established treatment. The demonstration of clinical superiority of the new product is certainly welcome here, but non-inferiority, showing that the new product is at least as good as established therapy, can be acceptable too. Also, admittedly, the size of the non-inferiority margin can be a matter of discussion or even controversy. A non-inferiority design requires that it is sufficiently certain that the established therapy, the one used as a comparator, has always a clinically relevant effect. Otherwise, a seemingly successful non-inferiority trial could be nothing but the finding that there is no difference between two groups without any beneficial effects in either group. Therefore, when comparators with weak or with uncertain efficacy are used in a non-inferiority trial, a three-arm design with the Firstly, new product, secondly, the comparator, and thirdly, a placebo arm may be the best option. In rare and complicated cases, we may have a situation with proven efficacy, superior to placebo, but inferiority to the comparator, to the standard of care. This may be acceptable if serious harm to patients can be excluded or can be avoided by the use of a rescue medicine. In other words, if loss of chance is not an issue. In that case, an alternative treatment option, even if less efficacious, can be offered, can have a value. But importantly, the situation would be quite different if we were to risk an irreversible progression of the disease by delaying the use of a more efficacious medicine. Loss of chance issue, we will come back to that.
We now leave efficacy and go to safety.
For the safety of medicines, data from preclinical studies, from the clinical development of the medicine and pharmacovigilance data collected after marketing of the product provides 3 lines of defense. Preclinical safety data are required for the start of the clinical development, another basis for the clinical trial authorization for the first-in-human studies. In addition, preclinical data will complement the safety data from the clinical trials. In particular, for adverse effects that cannot be studied or cannot be expected to be perceivable in clinical trials, such as carcinogenicity or teratogenicity. For the assessment of marketing authorization applications for the benefit-risk balance, safety data from the clinical trials are most important. As they frequently are from the very same patient as the efficacy results, they give a reasonable picture of the weight of risks and the weight of benefits. They are, however, of limited or of no use if the detection of rare or very rare adverse effects, which may become detectable only after huge numbers of patients, say 10,000 or more, have been exposed. Even when exceedingly rare, severe, or even life-threatening adverse events revealed by post-marketing pharmacovigilance activities may change a product's benefit-risk balance, may turn a benefit-risk balance negative and result in a suspension or revocation of the marketing authorization. Recognizing the importance of the post-marketing safety data, the European Union established the EMA's Pharmacovigilance Risk Assessment Committee, the PRAC, in 2012. Today, PRAC and CHMP work together to ensure that a positive benefit-risk balance is preserved over the life cycle of the medicine. We can be very short for the pharmaceutical quality. Frequently, we insist on improvements, but issues with pharmaceutical quality are almost never crucial grounds for the refusal of a marketing authorization for an innovative new medicine. Of course, this is different for generics. As efficacy and safety of a generic cannot be better than a reference product, there is no conceivable justification for potentially relevant differences in pharmaceutical quality. At variance for an innovative medicine, we will have the holistic view of all 3 criteria: efficacy, safety, and pharmaceutical quality. If the available quality of a medicine as used in the pivotal clinical trials resulted in benefits that outweighed the risk, why would we deny patients the benefits just because we think pharmaceutical quality should be even better? In such a situation, and if there's room for improvement, this can be dealt with after authorization in a quality variation. As emphasized earlier, marketing authorization is not an end in its own. A medicine, even with a positive benefit-risk balance, is never considered in isolation, but in its medical, societal, and economical context.
Regulators, and even more so downstream or subsequent decision makers, such as HTA bodies, payers, and, most importantly, patients and their physicians, will always judge the medicine and its efficacy against the expected normal clinical course of the medical condition or disease and against available treatments, against, if you like, competing products.
We can try to visualize this as a certain level of efficacy, low, medium, or high. indicate some range, differences between efficacy for individual patients, or some range of uncertainty on the magnitude of the efficacy of the medicine. At the bottom, we have the normal untreated clinical course of the disease. The possibility of a placebo effect is a reality and therefore reflected here as well.
In the gray area, we have usually old products, obsolescent or even obsolete products, authorized sometimes decades ago in the last century. And with them, we have their generics, which we authorize even today as generics. These medicines are efficacious. They do have pharmacological activity, but they are clearly inferior to what we would consider today's state-of-the-art treatment.
The state-of-the-art treatment is the current standard of care in the green zone. And there may well be different options, various products, even with some minor differences in efficacy, but still all considered acceptable treatment options, considered standard of care, even if one of them might be generally considered as the best of the best.
For a new product, CHMP will, after assessing the submitted material, conclude on efficacy as the first thing to look at. You remember, without efficacy, there's no way to a marketing authorization. Safety and pharmacological quality cannot save a development that failed to demonstrate efficacy. On this slide, we have the clear cases. If the new product is as good as the current standard of care, the first two small green lines, this is of course fine. And if the new product may possibly, just possibly be the new best, if we are sure it is at least as good as a standard of care, but maybe even the new best, of course, this is fine as well. The best possible outcome is when it is very clear that we do have a new standard of care and a new best, which is better than anything we had so far.
On the other hand, there are the red ones. If the new product does not do better than placebo, if we cannot be sure that it's better than placebo, it is not approvable. There have even been cases where the data was so limited that we had to say we simply don't know the efficacy. Could be average, could be good, could be very good, could even be the new best, but could as well have no efficacy at all. Without convincing evidence that there's a relevant efficacy at all, there is no way to approve a new product. In these cases, the final message for the applicant can only be, if it is really good, please produce the evidence, the data, and come back when you have it.
And then there are the tricky cases. Yes, there is undoubtedly pharmacological activity. There is undoubtedly some efficacy. But it is evidently or is probably or is possibly inferior to the standard of care. For example, what we do with products that are clearly efficacious, that do have on their own a positive benefit-risk balance, and yet are inferior to the standard of care?
A difficult one for the regulators. We need to ask the question, what does this mean for the patients? In particular, would the use of this inferior product result in a loss of chance? If the new product is less likely to work than the established treatment, would this cause irreversible damage, or could I give it a try? And if it does not work, switch to the established treatment as a rescue medication. If, for example, the treatment is for a mild to moderate pain, why not try the new product first? And if it does not work within an acceptable period of time, go in with ibuprofen, paracetamol, or whatever stronger painkiller makes sense. On the other hand, if the treatment is for a serious bacterial infection, say for septicemia or for a malignant tumor that may progress, we would not want to waste any time with a product that is less likely to work. For this product, we may have the question how much inferiority might be acceptable. While, of course, a non-inferiority trial against a standard of care comparator is acceptable, there will be a non-inferiority margin. Frequently, 15% have been considered acceptable. Would a higher inferiority margin be acceptable, or is even the 15% possibly too much?
This product now, that seems to be in the standard of care range, possibly even the new best, with some remaining small uncertainty that there might be a minor, say less than 15% inferiority. In such a case, the product is less a challenge for regulators than for health technology assessment bodies and payers.
For us, efficacy is obviously there. We even hope that post-marketing authorization, it might develop into a new standard, into a new best. Maybe a good situation for a conditional marketing authorization.
Here, we more often get in these uncomfortable situations that a medicine is authorized. The HMP even concludes that the product has a major therapeutic advantage, but HTA bodies and payers who make reimbursement and price decisions sees it differently. And therefore, the product will not become available for patients after all.
We are now coming to the end of this presentation, which should give you the first orientation of the regulatory world dealing with marketing authorization applications.
The ultimate goal, the overall objective, is the benefit for patients. And working on this, regulators have a double role in the marketing authorization of medicines. On the one hand, stopping bad medicines from damaging patients, the goalkeeper function. On the other hand, at the same time, we strive to clear the way for beneficial medicines, to facilitate the development from research to clinical routine. A marketing authorization cannot be an end in itself. The medicine must reach the patients. Remember the bridge we saw? All parts of the bridge are needed. Therefore, we work together we support upstream and downstream decision makers.
The European system is a network, with one part being the European Medicines Agency and the other part being the national member states with their national agencies. Both come together and collaborate in the scientific committees. For marketing authorizations, it is a Committee for Medicinal Products for Human Use, CHMP, whose opinions are the basis for decision of the European Commission. We have seen the composition of CHMP with delegates from all member states of the European Union and the European Economic Area, and how CHMP works, with rapporteurs and co-rapporteurs teams drafting the assessment, focusing on the benefit-risk balance. The procedure has at least 2, frequently more cycles, with other member states contributing and the applicants completing their initial submissions responding to CHMP's questions and objections. We gave a glimpse on the assessment of the benefit-risk balance based on the medicine's evidence for efficacy, safety, and pharmaceutical quality. There will be a dedicated presentation on benefit-risk assessment by Francesco Pignati, and we will come back to even more specific issues in the oncology part.
For us, this has been a pleasure, and we will be happy to answer any questions you may have right now.
Or we will be happy to receive any questions, comments, or feedback later at your convenience. Here, on the very last slide, you have our coordinates. Thank you.
And goodbye.